Assalamualaikum warahmatullahi wabarakatuh. Hello everybody. Before we start our lesson, let's pray together. Start. Enough. How are you today? I hope you are always in a good condition because it's a COVID-19 pandemic. You have to always maintain your healthy with the consume of vitamin C, protein, and carbohydrate. Now I will check a person today. Who's absent today? Oh, good. No one is absent today. Today we have to study about protein synthesis. Before we study, I have a question for you. When the high creatine protein is damaged, how does our body respond? Yeah, you can answer after you study about the protein synthesis. In this material, you will know how the process of forming new protein. Okay, now I will explain about past lesson, DNA and chromosome. This method can be used as a basis for a process of protein synthesis like that. DNA, or deoxyribonucleic acid, is the molecule that carries the genetic information in non-cellular forms of life and some viruses. It belongs to a class of molecules called the nucleic acid, which are polynucleotides, that is, long chains of nucleotides. Each nucleotide consists of three components. The first is nitrogen base, like cytosine, adenine, thymine, and guanine. And the second is 5-carbon sugar molecule, or deoxyribose in the case of DNA. And then the third is phosphate molecule. The backbone of the polynucleotide is a chain of sugar and phosphate molecules. Each of sugar groups in the sugar phosphate backbone is linked to one of the 4 nitrogen bases. DNA's ability to store and transmit information lies in the fact that it consists of 2 polynucleotide strands of twists around each other to form a double-strand helix. The base link across the 2 strands in a specific manner using hydrogen bonds. Cytosine pairs with guanine and adenine pairs with thymine. And then genes. The gene is the basic physical and functional unit heredity. It consists of a specific sequence of nucleotides at a given position on a given chromosome that codes for a specific protein, or in some cases, an RNA molecules. Gene consists of 3 types of nucleotide sequence. First is coding regions, called exons, which specify sequence of amino acids. And the second is non-coding regions, called introns, which do not specify amino acids. And the third, regulatory sequence, which play a role in determining when and where protein is made and how much is made. A human being has 20,000 to 25,000 genes located on 46 chromosomes, or 23 pairs. These genes are known collectively as the human genome. And then chromosomes. Eukaryotic chromosome. The label eukaryote is taken from the Greek for true nucleus, and eukaryotes is all organisms as viruses, bacteria, and archaea are defined by the possession of a nucleus and other membrane-bound cell organelles. The nucleus of each cell in our bodies contains approximately 1.8 meters of DNA in total, although its strength is less than 1 million centimeter thick. This DNA is tightly packed into structures called chromosomes, which consist long chains of DNA and associated proteins. In eukaryotes, DNA molecules are tightly wound around protein called histone protein, which provides structural support and play a role in controlling the activities of the genes. A strand 150 to 200 nucleotides long is wrapped twice around core of a histone proteins to form a structure called nucleosome. The histone dimer at the center of the nucleosome is formed from two units. Its histone H2A, H2B, H3, H4. The chains of histone are coiled in turn to form a solenoid, which is stabilized by the histone H1. Further coiling of the solenoids forms the structure of the chromosome proper. Each chromosome has P arm and Q arm. The P arm, from the French word petit, meaning small, is the short arm, and the Q arm, the next letter in the alphabet, is the long arm. In their replicated form, each chromosome consists of two chromatin. The chromosome and the DNA they contain are combined as a part of cell cycle and pass to the other cell through the process of autosomes and a pair of sex chromosomes. Two X-sex chromosomes for females, XX, and XY is sex chromosome for males. One member of each pair of chromosomes comes from mother, and member of each pair come from the father. A photograph of the chromosome in a cell is known as karyotype. The autosomes are numbered 1 until 22 in decreasing size order. Okay, after I explain about the DNA in chromosome, now I will demonstrate about the protein synthesis. You can take a note in your handout about the protein synthesis. Protein synthesis is a production of protein. Actually, there is a two process, transcription and translation. In eukaryote cells, transcription takes place in nucleus. During transcription, DNA is used as a template. to make molecule messenger mRNA. The molecule of mRNA then leaves the nucleus and goes to the ribosome in the cytoplasm, where translation occurs. During translation, the genetic code in mRNA read and used to make a protein. These two processes simplify the central dogma of molecular biology, DNA to RNA to protein. And then transcription is the first process of the central dogma of molecular biology. It's a transfer of genetic instruction in DNA to mRNA. During transcription, a strand of mRNA is made to complement a strand DNA, like that. And then, step of transcription. There are three steps: initiation, elongation, and then termination. Initiation is the beginning of transcription. It occurs when the enzyme of RNA polymerase binds to a region of a gene called promoter. The signals the DNA to unwind, so the enzyme reads the bases in one of the DNA strands. The enzyme is ready to make a strand of mRNA with a complementary sequence of bases. And the second is elongation, is the addition nucleolus to mRNA strand. The third is termination, is ending of transcription. The mRNA strand is complete and detached from DNA. Okay, now let's move to processing mRNA. In eukaryotes, the new mRNA is not yet ready for translation. At this stage, it's called pre-mRNA, and it must go through more processing before it leaves the nucleus as mature mRNA. The processing might include splicing, editing, and polyadenylation. The process modifies the mRNA in various ways. Such modifications allow a single gene to be used to make more than one protein. Splicing. Splicing removes introns from mRNA. Introns, a region to do not code for the protein. The remaining mRNA consists only of regions called exons that do code for the protein. The ribonucleoprotein in the diagrams are small proteins in the nucleus that contain RNA and are needed for the splicing process. Editing. Editing is change some of nucleotides in mRNA. For example, a human protein called ApoB, which helps transport lipids and the blood, has two different forms because of editing. One form is smaller than the other, because editing adds an earlier stop signals in mRNA. And then polyadenylation adds a tail to the mRNA. The tail consists of adenine base. It signals the end of mRNA. It's also involved in exporting mRNA from the nucleus, and it protects mRNA from enzymes that might break it down. Let's move to the translation. Translation is the second part of the central dogma of molecular biology. It's process which the genetic code in mRNA is read to make a protein. After mRNA leaves the nucleus, it moves to a ribosome, which consists of rRNA and protein. The ribosome reads sequence of codon in mRNA and molecules of tRNA bring amino acid to the ribosome in correct sequence. To understand the role of tRNA, you need to know more about its structure. Each tRNA molecule has an anticodon of the amino acid it carries. An anticodon is complementary to the codon for an amino acid. For example, the amino acid lysine has the codon AAG, so the anticodon is UUC. Therefore, lysine will be carried by a tRNA molecule with anticodon UUC. Wherever the codon AAG appears in mRNA, a UUC anticodon of tRNA temporarily binds. While bound to mRNA, tRNA gives up its amino acid. With the help of rRNA, bonds from between the amino acid as they are brought one by one of the ribosome, creating a polypeptide chain. The chain of amino acid keeps growing until a stop codon is reached. So what's happened next? After a polypeptide chain is synthesized, it may undergo additional process. For example, it may assume a folded shape due to interaction between its amino acids. It may also bind with other polypeptides or with different types of molecules such as lipid or carbohydrates. Many proteins travel to the apparatus Golgi apparatus within the cytoplasm to be modified for a specific job they will do. Okay, after I explain about the protein synthesis, let's see your student worksheet. There are two questions. Question number 1. Based on your discussion with your friend, complete the empty box of protein synthesis below. There are 5 empty boxes. 1, 2, 3, 4, and 5. You can complete the empty box. You can analyze the box that is full of DNA, ribosome, and protein. Okay, number 2. After complete the empty box, analyze the process that occur and write down the explanation about the vocabulary. You can write down. You can look in the number 1 for fulfill in the box number 2. And the last box is for your reference. You can write your reference from internet or articles and books in this box. Are you understand? It's easy for you, I think. I give 20 minutes to do it. Let's discuss with your group. Good luck. Okay, I forget, but if you have difficulties to do student worksheet, you can ask me a question about it. Are you understand? Good. Okay, let's discuss about your answer of worksheet I have done. The first is number 1. Number 1 is complete the box the empty box. There are 5 numbers. Point 1. Point 1 is transcription. And then point 2 is RNA messenger. Point 3 is translation. Point 4 amino acid, and then 5 is transfer RNA or tRNA. Okay, let's move to number 2. Point 1 and point 2. I want to Alif, please share your answer of point 1 and 2. Alif, please.
Okay, Miss. The answer of number 1 is transcription. Takes place in the 3 steps: initiation, elongation, and termination. For the step is initiation. Initiation is the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. This signals the DNA to unwind so the enzyme can read the bases in one of the DNA strands. The enzyme is ready to make a strand of mRNA with a complementary sequence of bases. Second step is elongation. Elongation is the addition of nucleotides to the mRNA strand. And the last step is termination. Termination is the ending of transcription. The mRNA strand is complete and it detached from DNA. Next, for the answer of number 2 is RNA messenger. In eukaryotes, the new mRNA is not yet ready for translation. At this stage, it is called pre-mRNA, and it must go through more processing before it leaves the nucleus as mature mRNA. The processing may include splicing, editing, and polyadenylation. These processes modify the mRNA in various ways. Such modifications allow a single gene to be used to make more than one protein. For the first is splicing, removes intron from mRNA as shown in the diagram below. Introns are regions that do not code for the protein. The remaining mRNA consists only of regions called exons that do code for the protein. The ribonucleoproteins in the diagram are small proteins in the nucleus that contain RNA and are needed for the splicing process. For the second one is editing. Editing changes some of the nucleotides in mRNA. For example, a human protein called ApoB, which helps transport lipids in the blood, has two different forms because of editing. One form is smaller than other because editing adds an earlier stop signal in mRNA. And the last one is polyadenylation. Polyadenylation adds a tail to the mRNA. The tail consists of a string of S or adenine bases. It signals the end of mRNA. It is also involved in exporting mRNA from the nucleus, and it protects mRNA from enzymes that might break it down.
Good, it's true. Like more demonstration about the protein synthesis. Now let's answer the number 2.3, 4, and 5. Yeah. Number 3. Number 3 is translation because ribosome to protein. So the process is translations. The answer number 3 is translation occurs and the ribosome, which consists of RNA and proteins. In translation, in instruction mRNA are read and tRNA brings the correct sequence of amino acids to the ribosome. Then, rRNA helps bonds from between the amino acids, producing a polypeptide chain. Okay? I understand? In U2, the score is 5. And then number 4, box 4 is amino acids. Box 4 is amino acids. Amino acids are the results of translating codons by ribosomes. Amino acids will be formed into proteins. Its score is 5-2. If you true, and then number 5. The empty box of number 5 is tRNA. Yeah, transfer RNA is the molecules of RNA that serve to bring amino acids to a growing polypeptide strand and properly place them into the sequence. The score is 5. Yeah. The final score is your total score per 40 or max score times 100. You can get your final score and write down in your student worksheet. Okay. Okay. You have to finish it. After it, I have a question for you about a summary of this lesson today. Intan, please share about summary this lesson today. Please, Intan, share with me and your friends.
Yes, Miss. Summary of protein synthesis topic are: number 1, protein synthesis is the process in which cells make proteins. It occurs in 2 stages, transcription and translation. Number 2, transcription is the transfer of genetic instruction in DNA to mRNA in the nucleus. It includes 3 steps: initiation, elongation, and termination. After the mRNA is processed, it carries the instruction to a ribosome into cytoplasm. Number 3, translation occurs at the ribosome, which consists of rRNA and proteins. In translation, the instruction in mRNA are read, and tRNA brings the correct sequence of amino acids to the ribosome. Then, rRNA helps bonds form between the amino acids, producing a polypeptide chain. Number 4, after a polypeptide chain is synthesized, it may undergo additional processing to form the finished protein.
Okay, good, Intan. Excellent, very good. Thank you, Intan. I have a video for you. Watching this video will improve your understanding about the protein synthesis. Okay, let's watching. Here is cell, the basic unit of a living tissue. In mushroom cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instruction for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. This messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosomes read the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acids. Transfer RNA molecules carry the amino acids to the ribosomes. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein. After watching the video, you will be able to make a video play a role of a protein synthesis. You have submit in next week via email, my email. And then thank you for your attention. Before we go home, let's pray together. Start. Enough. Okay? See you next time and wassalamualaikum warahmatullahi wabarakatuh.